Let's go on down with our check-in today. How about Ashton? We'll have you go first. Cookies or cake and what
probably
kind?
Probably chocolate chip cookies.
Awesome. I'm sorry, I didn't mean to interrupt you. Good choice. So he's going old school from the start, right? How about Connor?
I would do the same thing, chocolate cookies.
Okay. All right, Aaron, how about you?
Definitely cookies over cake for sure.
Okay.
And I'd have to go with peanut butter cookies.
All right. Very good. Good choices, you guys. Nathan, what would you pick?
Cake over cookies and angel food cake.
Ooh, angel food cake. Nice. Okay, how about Danner? I think he's not in yet, huh? That's not normal for him. Okay, how about, do we have Ariel?
I would do the same with Ashton.
The question was cookies or cake and what kind?
I would do the same as Ashton.
Oh, the same as Ashton. That's what you said. I'm sorry. Okay, very good. Okay, how about we move on to, let's see. Hey, Alex, what would you pick?
I'd go definitely with red velvet cake.
Ah, red velvet cake. All right. How about Leah? Look at the timing. She just came in. We couldn't have paid her to do that. Leah, the question is cookies or cake and what kind?
I would say cookies, but I also really like cake. But I would take a just simple chocolate chip cookie.
Nice. And then what cake would you pick?
Oh, that's a tough one. Probably the Costco cake. I love their cake. It's so good.
Yeah, they do. Man, don't they do that well? All right, I agree. Amanda, cookies or cake?
I'd say cookies. And it's weird, but I like oatmeal raisin.
Nice. So does my daughter. That's nice. Good choice. Okay. How about Ashley?
Chocolate cake.
All right, chocolate cake. Nice. Costco cake, or have you had their chocolate cake?
I just like any chocolate cakes.
Yeah, any works. Good choice. Okay. How about Ryan?
I would take chocolate chip cookies.
Very good. Thank you. Okay. How about, let's see. How about Amber?
I would say cookies, chocolate chip.
Chocolate chip cookies. Okay. Damien.
Peanut butter cookie.
You said cookie?
A peanut butter cookie.
Nice. Peanut butter cookies. Those are great too. Okay. How about then Miranda? Hey.
I would choose an M&M cookie.
Nice. M&M cookies. Good. Okay, Kylie? Do we have her yet? Yeah, there she is. Nice. Very good. Thank you. How about Grace?
I would choose just plain chocolate chip cookies.
Plain chocolate chip. Nice and simple. Good. Let's see. Do we have Liam yet? I don't think so. How about then Jasmine? Is she? I don't think she's here yet either. I don't see the purple. Okay. How about Aiden?
I'm gonna have to go with like oatmeal raisin cookies.
Good. Oatmeal raisin cookies. Nice. Okay. I'm gonna take a quick scan. Okay. And then, did we get Danner at all? I don't see Robert either. Okay, that's fine. So what I want to do is kind of wrap up some of our notes dealing with polygenic traits. We really haven't addressed that fully, and that won't take long. and then we're going to get into pedigrees. So I would think it would help if I share my screen with you on that. So I'm going to pull that up for us. Anybody have something fun planned for this week? Not yet. Hey, Danner. Okay, now let's make sure you can actually see the screen. Okay. Can you see the screen that says incomplete dominance?
Yeah.
Yeah, okay, thanks. Okay, I'm going to jump down to the end because that's where we are. So for polygenic traits, we've touched upon this a little bit in the past. So poly means what? What's that prefix mean?
Many.
Thanks, guys. Yes, right. Many. Very good. And then genic probably reminds you of genes, right? So we've got certain traits that actually are controlled by multiple genes. So this poly means many, and genic sounds like genes. Okay. So this is an inheritance pattern controlled by 2 or more genes. Now, the interesting thing about this is that the genes can be on the same chromosome, or they can actually be on different chromosomes and still be polygenic, still be related. So, you know how we usually write genotypes, the traditional naming system, like big A, big A, big A, little a, and then little a, little a. Well, when you're talking about polygenic, then you're going to just use different letters for each gene. Like maybe the second gene could be big B, big B, big B, or little b, little b. And then if you're talking about something that has a third gene, maybe that third gene is big C, big C, little c, or little c, little c. So we don't have to get too complicated with that. Just, you know, simple A, B, C, D, and so on. Now, skin color actually is a classic polygenic trait, and so is your eye color, because it's just dealing with pigment, right? So skin color is influenced by the additive effects of melanin, and it can be up to 3 to 6 genes that control your skin color, which accounts for all the tremendous variety in skin color. Now, when we talk about skin color, obviously there's environmental influences, right? So if you go outside and tan, your skin's going to change color, but we're ignoring environmental factors in this scenario and just focusing on genes. Now, the amount of melanin or pigment in your eye color is also controlled by multiple genes. So that's why we've got such different shades and great colors in eye as well. Are you with me so far on this? Thumbs up if I'm making sense. Here, you already lost me today. Okay, so, all right. So let's take a look at this graph here. And this is an example focusing on skin color. So what they did in the P generation, took the lightest genotype possible where every one of those 3 genes is recessive. And they crossed it with the opposite. Every one of those 3 genes is homozygous dominant, right? They're homozygous dominant here. And these are homozygous recessive. Okay, so that means when you do the F1 generation, everybody is heterozygous, right? We're all hets. Heterozygous. So if you take the F1 generation that's strictly heterozygous and cross it with another heterozygous, you get this huge variety of skin types, right? So in the beginning of the genetics unit, we crossed 1 gene. So there's this formula, 2 to the nth power, where the n is the number of genes. N is the number of genes. So if you have 2 to the first power, you're studying one gene, and you get 2 to the first power is 2, so your Punnett square ends up being 2 by 2, right? If we're studying one gene, you're going to get a little 2 by 2 Punnett square. Now, when we added 2 traits, like the dihybrid cross, 2 to the second power is 4. So then you saw us doing Punnett squares that were 4 by 4, right? And now here, we're using a cross that's 2 to the third power, which is 8. And that's why you see a cross that's 8 by 8 now. Okay. And the pattern would continue, right? If you're going to do a cross with 4 traits, that's 2 to the fourth power. And then if we were going to talk about 5 traits, that'd be 2 to the fifth. And you can see if we go to 6 traits, these crosses are getting gigantic, right? So if you take a look and do this cross, this is actually the ratio for the phenotypes down here, right? Not even the genotypes. They didn't even bother listing the genotypes because that's just now an ugly mess. So the most common one you see in all 64 crosses, 20 of them are going to be this skin type. And the least common are this super dark and super light. And you can see those at the end, the extremes of the bell-shaped curve. So when you study pigments for skin color, you always get this bell-shaped curve, and that's why you get the bell-shaped curve. We figure that out based on our Punnett square. Does that make sense? Okay, you're good? And then the other thing you can also study in polygenic traits are things like pyloric stenosis. The word stenosis, whoops. If somebody says they're studying a part of your heart and it's stenotic, stenosis means narrowing. And pyloric refers to your digestive system. So in this case, when we study pyloric stenosis, it's the narrowing of a passage that connects the stomach to the intestines. So we're talking about a part down here that's narrow. And I'm sure you can agree that is pretty dangerous. That's not something that you want, narrowing, okay? Because otherwise you're gonna have issues like if you can't empty that stomach of food, you're gonna end up with this starting to rot with bacteria and stuff, right? And if that can't empty properly, you risk things like sepsis. I don't know if you've heard the term septicemia, okay? So that's, you know, an infection that's caused many different ways, not just this way, okay? But it's a bacterial infection. Another polygenic trait is cleft lip and cleft palates. So you can see examples down here. The interesting thing and the fascinating thing to me is now doctors have gotten so good at repairing this. I've had students that had severe cleft lip or cleft palate, and you'd never even know looking at them today. And the issue with this is if they can't close their mouths or the palate is deformed, that means as a baby, they could not nurse properly, whether you're talking about breastfeeding or bottle feeding. And if they can't get the nutrients, obviously, that has some pretty negative consequences. That can be very dangerous. How many of you have heard of cleft palate? Does anyone know some who had that by chance? Okay. So
I know a celebrity who did.
Who?
Joaquin Phoenix.
Oh, that's right. That explains the scar on his lip there,
Yeah.
right? That's pretty cool. Cool. I just think it's so cool that we can help these babies, and for the most part, you might have no idea. You know, there's no effect. Maybe you see a scar, but and then there's spinal bifida. Raise your hand if you've ever heard of spinal bifida. Okay, so this is another birth defect, and this picture shows really well. What happens is the spinal column doesn't form properly, and you're getting parts of the spinal cord and nerves that are actually outside of the spinal cord. So there's literally this area on the outside of the body, and obviously you know you can't afford to have that injured in any way, right? So what doctors usually do is try to perform surgery and close that in a protected way. And then here's club feet. That's another polygenic trait. You can see why it gets that name. Okay, lots of great surgeons out there too do a great job at fixing that as well. I don't think you can get quite as close to perfect as you can in a cleft palate surgery, but I'm not claiming to be an expert on that. You're going to have to look into that further if you're interested. Questions on these? So that's why we study polygenic traits. It's fun to look at eye and skin color, but here it's also important to see what happens or what could go wrong when you're dealing with polygenic traits. Now, who remembers what a karyotype is? Okay, Grace, can you jog our memory?
It's like putting the chromosomes together to see what disabilities or like genetic disabilities the person would have according to their chromosomes.
Very good. So don't forget, when you talk about chromosomes, I want you to think of them as cooked spaghetti noodles or maybe a worm. They're all floppy, okay? So just because this one's arranged this way and it's kind of curved doesn't mean that in another way it wouldn't be much straighter. So when you look at the chromosomes, you can study to see if they have extra chromosomes or extra parts of chromosomes. So just like you have genetic abnormalities, these will help you study chromosome or abnormalities. It will not help you identify like a mutated gene. This focuses on the chromosomes, not the genes. And you sort them based on 3 things. You pair them based on 3 things. 1 is the length of the chromosome. Another one is the banding pattern, that striping pattern. You can see the banding patterns, say of pair 1, are very similar to each other. And the third thing that you use to pair them up is the position of the centromere. Remember, the centromere was what we talked about in mitosis and meiosis, where those spindle fibers connect to the sister chromatids, right? So you'd study where those positions are. The centromere isn't always in the dead center. It can be above center towards the top. It can be below, and all sorts of varieties. So you can see here, this karyotype, is this male or female? Give me a 1 for male, 2 for female. Male, 1, 2, female. This is a female. You look here, she has 2 X chromosomes and no Y chromosome. So that's how we know she's a female. But here you can see male or female. 1 male, 2 female. This is a male, absolutely. Good. So you might have a situation where you see somebody and they have an extra of chromosome of pair 21. Do you remember what that's called?
Downs? Like Down syndrome?
Exactly. Yep. Down syndrome is called trisomy 21 because you have 3 chromosomes for pair 21 when you should only have 2. Okay. So it's important to look at genetic disorders and chromosomal disorders. Questions on that? No? Okay, so let's say you want to study the passage of a certain genetic disorder. Well, that's when pedigrees come into place. So I'm going to close this file. I have a different one for us. All right, so I have a pedigree PowerPoint here, and these slides are also on Canvas for you. Okay, so when you use a pedigree, you're really studying the family tree of somebody's genes. So there's certain, what do you guys remember about pedigrees? Have you done them before, maybe? Raise your hand if you've done something with them before. Okay, Ariel, you have? The rest of you have never done a pedigree? Amanda, do you remember what you did with it?
I think I have, and is it like a family tree?
Yes.
You start with like the grandparents and then you go down?
Yeah, let me show you a picture.
Yeah.
Maybe that's, hey
Yeah,
guys, do you remember studying something? Yeah, sorry. Guys, so how about the rest of you? Have you done anything with an image that looks kind of like this? I know Ariel, yep. And then do you remember at all, Ariel, what you were doing with it?
Worked with them in biomed.
Oh, yeah, that's right. Biomed, of course. So yeah, so the pedigree is a chart showing genetic history among several generations. In college, one of my senior projects, I had to do a pedigree. And I think we had to do like, I don't know, 3 or 4 traits. So when I became a bio teacher, I wanted to do that activity with my students because I thought it was really fun. And surprise, I couldn't find my old lab directions. So I contacted my genetics professor and said, hey, that was a really good assignment. I want to do that with my high school students. And she said, I'll give you the assignment, but I want you to know I don't do that assignment anymore. So of course I said, well, how come? Why don't you do that assignment? And she said, well, one year I was doing that pedigree assignment and turns out one of my students hadn't been told she was adopted. So when she went to do the project I assigned her, dun, 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 you know what happened. She figured that one out. And apparently that had been a family secret that she, inadvertently discovered. She had no idea. They had never told her for whatever reason. So she said, yeah, so that's why I don't do that assignment anymore. So you can find out a lot of very interesting stuff doing something as simple as a pedigree. Okay, so a scientist or a genetic counselor might look at your family history and use a pedigree, say if you're concerned about like passing a certain trait on, like there's one that's coming to mind, oh, Huntington's disease. Have you guys ever heard of Huntington's disease? It's actually a dominant trait, a disease, and you don't realize you have it usually until you're in your 40s, unless there's been a clear family history of it. And the problem with that is it's a dominant disorder that is pretty awful, an awful way to die. And think about it, if you don't figure out you have this disorder that you could pass on your kids until your 40s, well, most people have kids before they're in their 40s, right? So you could have already inadvertently passed that disorder, that disease on to somebody without knowing you had it in the first place. So just a quick review to jog your memory. Remember, females are circles. So the memory trick I use is females are typically curvy compared to males. So that's why the females get the circles, and then you goofball males, you guys get the squares, right? But you don't have to give birth, so I think that's a pretty easy trade-off for you guys, right? Okay, all right. So I'm going to show you a screen. This is going to be part of your homework for tonight, so we're going to get started on it together if that's okay. All right. Now I'm going to warn you a little side note. I'm being a little bit dangerous today, and my battery power says I have a half hour left. So if I cut out of the middle of nowhere, it means my computer died, okay? So I have to get the cord. I had obviously unplugged it. So anyway, why don't I give you a moment to go in Canvas, and I'll show you how to access this assignment, because we might as well work on it directly so you don't have to go back and make more work. So when you're in Bio for Canvas, you're going to scroll down to our genetics unit. Okay. And then you should see pedigree practice. It's due May 13th. It says 0 points. I'll have to fix that later. All right. So then if you just open that document up, it's going to look like this. Come on, squirrel. Okay, this is what yours looks like if you want to take a moment to open it up. Give me a thumbs up when you have yours pulled up. Do you guys have it? Okay. Ashton, do you need more time to pull that up? You got it? Okay. I can't see your face, Ashton. Show me. I hope you have clothes on. Tell me you have clothes on, Ashton. Uh-oh. He says he hasn't said anything, so I'm assuming that's a no comment. Yikes. Okay, so for pedigrees.
No, I have clothes on.
Okay, good, good. That's a good way to start the Monday off, at least, right? Okay, yeah. So pedigree is a visual chart that depicts the family history or transmission of a certain trait. They can be interesting to view and important tools in determining patterns of inheritance for specific traits. Pedigrees are used primarily by genetic counselors when helping couples decide to have children when there is evidence of a genetically inherited disorder in one or both families. Like I was talking about Huntington's disease. They're used when trying to determine the predisposition of someone carrying a hereditary disease like familial breast cancer. For breast cancers, there's different forms of breast cancer, just so you know. Some of them are genetic and others are not. Okay, so be aware if somebody's diagnosed with breast cancer, that doesn't automatically mean it's genetic. Okay, because there's hormonal things that go on as you age that can cause breast cancer that would not be genetic. So we talked about the circles and the squares. The vertical line represents parents and their children. The horizontal line connects them in some sort of relationship. That used to mean that it was marriage, but not necessarily true these days. Shaded circles or shaded squares mean they express the trait. They have the trait you're studying. Okay. If it is not shaded, it means they have not expressed the trait. Now, I want to be very clear when I say this. If they have the trait, the full shading will already be done for you. But if they're a carrier, somebody who could pass that trait on but doesn't suffer from it, you and I are the ones who are gonna shade. So carriers, let me write that down. sorry, excuse me, carriers are ones that you would half shade, okay? Okay, and I'll show you how to do that as we approach examples like that. Okay. So here are just some basic notes on it. We talked about the circles and the squares. If the individual is affected by whatever you're studying, this is important, you're going to know that it's fully shaded for you. Okay. It's already darkened or shaded for you. Okay. Sometimes you see some shapes that are filled in halfway, and that means they're heterozygous or a carrier. And I'm going to star that because a lot of times you need to do that. Maybe it's not done for you. Okay. So let's start looking at some examples. It says, a pedigree is just like a family tree, except it's focusing on one specific trait. The pedigree usually only shows the phenotype of each family member. With a little thought and hints below, you can determine the genotype of each family member as well, usually. Okay, so there's a couple things I want to add to this in a moment. So hints for analyzing pedigrees. If the individual is homozygous recessive, both parents have to have at least one recessive allele, meaning the parents are heterozygous or they're homozygous recessive. And if you're like, I don't get what that statement means, when we start working on the pedigrees, I think that'll make sense. If the individual is dominant, then at least one of the parents has to be affected. Okay. If the offspring has a dominant trait, they must have gotten that from somebody, meaning they've gotten it from either mom or dad or both. You'll never see a dominant trait in a child without at least one parent being dominant. Okay, but you can see a child who is recessive and the parents aren't recessive. That means they're usually heterozygous. So they've got that trait, but they have the dominant allele getting expressed. Note in a pedigree, traits of interest can be dominant or recessive. The majority of harmful genetic conditions are only seen when the individual is homozygous recessive. Examples of conditions caused by recessive alleles include cystic fibrosis. That's a disease in your secretory glands, including those that make mucus and sweat. So you're getting filled up with mucus all the time. There's something called Fanconi anemia. It's a blood disorder where you don't have the proper red blood cells. And then I'm going to move real quick because I just saw the screen darken, and I'm going to go get my plug. So hang on. I can't find it. Yeah, here we go. You guys are still with me, it looks like, so that's good. Albinism means you have lack of pigmentation. There's a disorder called PKU, phenylketonuria. That's a metabolic disorder. And if you look at your soda cans, a lot of times in other foods, they'll have food warnings regarding people who have phenylketonuria. They call it PKU for short. It's a metabolic disorder where you can't break down certain substances. And if you fill with those substances, it'll cause brain damage. Some genetic conditions are caused by dominant alleles. And common examples, we mentioned polydactyly, where you have extra fingers. Achondroplasia, remember there used to be a show on cable, something about the little people? Does that ring a bell? Does that kind of ring a bell? Something about little people, and they were all short. That's actually the scientific term for that. Instead of dwarfism, it's achondroplasia. And then neurofibromatosis is a genetic nerve disorder. Then there's something in biomed, if you're with me, we studied familial hypercholesterolemia, and that affects your heart disease, and you have really high cholesterol levels. And if you're a fan of watching Pimple Popper, like I am, you'll see like people who have familial hypercholesterolemia, for some reason, they'll have like these really big deposits on their eyelids and on other body parts of cholesterol. And to somebody who knows about that background, that's like an indicator. If you have a patient and you're concerned about that, a lot of times you'll see these certain structures on their eyes and on the back of their feet and in their eyeball, not just on the eyelids. So I find it really fascinating. Danner, I saw you just yawn. Obviously, you didn't find that as fascinating as I do. Okay. You had a hard weekend too, right? This lousy weather doesn't help. So anyway, let's get going on this. So use the pedigree chart shown below and label. Some of the labels may be used more than once. So I'm going to give you a moment and I want you to answer questions 1 through 7 now. Okay? So answer questions 1 through 7 now, please. Does anybody need more time? Raise your hand if you do. Okay, how about Damien? Help me out with number 1. Which shape represents a male? What letter?
The square.
The square does, so that's letter C. Very good. Okay, and then how about Nathan? What about a female letter? Yep, A. Oops, that doesn't look like an A. Hey, Grace, how about number 3? Which one represents marriage?
That be
Yeah, that's D. Very good. Or I'm sorry, you said B, right? B represents marriage. How about Leah, number 4? A person who expresses the trait, that's letter
A.
Good. A is right. And then 5, Alex, a person who does not express the trait.
C.
Good job. Thank you. Okay. And then how about Liam, number 6, a connection between parents and offspring. Austin, can you help him out, please? I didn't hear him.
Would it be D?
D is right. Very good. Okay, Aaron, how many generations?
3.
Yeah, you should see 3 generations. Very good. Okay, I'm kind of flipping back and forth between screens. Okay, very good. That's letter D. Now, it says, assume the chart above is tracing the dominant trait called white forelock, okay, with a big F through the family. Big F is a tuft of white hair on the forehead. So before we answer those questions, since this is our first pedigree, I wanted to work out as many of the genotypes as possible. Now, we said it's dominant, right? So anyone who is affected must have at least one big F. So this is kind of like when you do puzzles, like jigsaw puzzles, you always do the easy stuff first. And if you're like me, the easy stuff for me is doing the edges first. And then I hop around to the harder stuff. So right where anybody who's shaded, you're going to want to write big F for their first letter. Okay. So if you are big F, then it has to be shaded, but we're not quite sure yet what the second F is. The second F could be big F, and you might be able to determine that with enough information. Sometimes you figure out that it's big F, little f, and sometimes it can be big F, and you have no idea what the second F is, so you'll write big F question mark. Does that part make sense? Okay. Is everybody still with me?
Hey Sharon, can you say that again really quick so I make sure that my notes are right?
Yes, thanks. So if we're talking about a dominant trait, and then everything, you'll see the shape that's shaded, and that tells us they at least have to have one big F. So that's why I went around and labeled everybody big F. But the second F we have to figure out. Option one, maybe that person ends up being big F, big F. Or they could be big F, little f. Sometimes there's not enough information to figure out what that second F is, so you write big F question mark. Does that make sense? All right, so let's start. Remember we said it's a puzzle, so let's do some easy stuff first. If this is dominant, then everybody who's not shaded is what two letters?
Little f?
Exactly, they have to be little f, little f. So right from the get-go, once you figure that out, I would label everybody who's not shaded little f, little f. This guy's little f, this one's little f, little f, little f, little f. Because remember, if they were big F, they'd have to be fully shaded. And I'll give you a hint. In this case, if it's a dominant trait, you can't even have a carrier that you half shade in. You only get carriers if the trait is recessive. Okay, so again, it's a dominant trait. So we know the ones that aren't filled in have to be homozygous recessive, little f, little f. Because the only way to be unaffected is to have two of the recessive alleles. So the next step, what we want to do is now hop around and see if we can figure out what that second F is. So I'm going to go look at this parent first at the very top. You don't have to go in order. Okay. Now look at their offspring. They had 1, 2, 3, 4 kids. 2 of them are affected and 2 of them are not. 2 of them are little f, little f. They had to get that little f from dad, and they had to get one from mom. So that means mom up here must be heterozygous. She has to be big F, little f. Does that make sense? Now I'm down to one screen, so I can't see your faces. So I need you to speak up if you're having trouble. Are you good with that, everybody? Okay, so then you would work through the rest of the ones. This parent or this child from these two people have at least one big F because it's shaded. And by the same logic, dad would have given this daughter a little f. So she too is heterozygous. Does that make sense? And that means that this son has the same scenario. So I don't see any case in which any of these shaded people would be big F in this example. You will see other examples where they are big F. Is that making sense? Okay, so we're almost out of time. So what I want you to do is, my advice to you would be to work out these in pencil so it's easy to erase. I want you to jump to the one that says example number 1. This is autosomal recessive. So that means to be shaded, it's the opposite. That means Arlene has to be little f. Anywhere they're shaded, they're now little f because the disorder is recessive. Are you with me on that? Danner, am I making sense? Parents, is that making sense?
Yeah.
Okay, good. Okay, I'm just gonna give you a few more hints so that you can go off and do the homework. Okay, so going back to this one, you will find offspring that are F question mark. They wrote it as F dash. I write this as F question mark because you can't tell what that second letter is. For example, Alan, if you look at Alan, he came from Tom and Sandra. Sandra had to have been a big F. And then if you look, she was married into the family. So we don't know what her second letter is at this point. They had a child who is heterozygous. And that little f came from dad. Okay. Now that big F must have come from Sandra. Okay. But that still doesn't give us enough information to be sure of what Sandra's second F is. So you're going to write her as big F question mark. Okay. When you look at Alan, he for sure got a big F, but he only could have gotten a little f from dad. So not only is Alan heterozygous, you have to half shade him because he's a carrier. Okay. And in this case, Tina married into the family. We know she must be a big F, but we don't know what her second allele is. So she's also going to be a big F question mark. So your options when you work this one out, and if it's recessive, remember you could be big F if you have enough information. You could be big F little f, or you could be big F question mark. So for your homework, what I want you to do is to finish that packet. You're going to deal with example 2, which is autosomal dominant, kind of like the first one we started. So the example 1 and 2 are going to work in an opposite manner because one's recessive and example 2 is dominant. Do you guys have questions? Okay, if you're good and you don't have questions, then you can head out and have a great day. Don't forget, I'll be here from 1 to 2 tomorrow, every Tuesday and Thursday, to Zoom if you need help or you email me. Have a great day, you guys.
Bye, Michelle.
Bye, guys.
Thank you. Bye.
You're welcome. Bye.